Today, we are going to talk about nucleic acid in details,
which include three major subtopics. They are the DNA replication, DNA transcription, and finally, the translation.
I'm Phil.
I'm Sunny.
I'm Isla, and we hope this video is helpful too.
So I'm going to talk about the DNA replication. DNA replication is required when the cells divide or replicate. The identical copies of DNA is made as the product of the DNA replication.
The Hershey-Chase experiment providing evidence for DNA is a genetic material rather than protein, and they used the feature of T2 that the protein codes of T2 virus only contains sulfur, while the DNA only contains the phosphorus. And the experiment cultured two groups of T2 bacteriophage, and one group of bacteriophage was cultured in the radioactive sulfur and another group cultured in the radioactive phosphorus. And after the T2 bacteriophage injected their genetic material into the bacteria, and they were centrifuged to concentrate the cell in the pellet, and the non-genetic material of the virus remained in the supernatant. And the result is that the radioactive sulfur was found in the supernatant and the radioactive phosphorus was found in the pellet. The first stage of the DNA replication is that the enzyme called helicase unwinds two strands of DNA, and the replication fork formed. And then the single-strand binding protein bind with each strand to keep them separated during the replication. And the strand from 3' to 5' is called the leading strand, and the strand from 5' to 3' is called the lagging strand. And the premise adds a short section of RNA primer on the leading strand. And then the DNA polymerase III starts adding complementary bases on the leading strand continuously towards the replication fork to form a new strand. However, on the lagging strand, after the premise adds RNA primer on the strand, the DNA polymerase III adds complementary bases on the lagging strand opposite to the replication fork discontinuously to form sections called Okazaki fragments. And then the DNA polymerase I removes the RNA primer with nucleotides, and after that, the ligase connects the Okazaki fragments together to form a new strand. There are some regions of DNA do not code for proteins are called non-coding regions, but they have other important functions. For example, the area of repetitive sequences occurs on the ends of the eukaryotic chromosomes called telomeres. The telomere serves a protective function. During the interphase, the enzymes that replicate DNA cannot continue replication all the way to the end of the chromosome. If the cells went through the cell cycle without telomeres, they would lose the genes at the end of the chromosomes. Sacrificing the repetitive sequences found in the telomeres serves a protective function.
Transcription is the process of transcribing the leading strand, or sometimes called antisense strand, by action of RNA polymerase. And it is required because the DNA is stored in the nucleus of the eukaryotes, and it need to exit the nucleus to initiate the next step, translation or protein synthesis. And mRNA is the section transcribed, and it is complementary to the original antisense strand.
RNA polymerase first unwinds the two strands of DNA, and the RNA polymerase also assembles the free nucleotide onto the antisense strand to form mRNA. And the mRNA actually contains the same genetic information as the sense strand, because antisense strand is complementary to the sense strand, and antisense strand is a template of the synthesis of mRNA. So mRNA, as the genetic information that is replicated, goes out of the nucleus into the ribosome to initiate translation. Gene expression is regulated by some specific proteins binding to the specific base sequences on the DNA. And for example, the promoter. The promoter initiates a transcription when RNA polymerase binds to it. And RNA polymerase is protein. And other examples could include silencers and enhancers. Silencers decrease the rate of transcription, and the enhancers increase the rate of transcription when certain proteins bind to it. And gene expression could also be regulated by environment. And for example, in E. coli, there are a section of genes responsible for lactase production. When there's no lactose in the environment, the repressor before the section of gene is activated, so it blocks the transcription of this section of gene. When there is lactose in the environment, the repressor deactivates and it enables the transcription of the section of gene. Environment could also affect the gene expression, but because the organisms adapt to the environment by changing the expression of the gene. And some genes are activated and some genes are deactivated in certain organisms. And it is done by the adding functional groups to the certain part of the gene or the histone. And the gene or DNA is wrapped around the histone and forming a complex called nucleosome. For example, the acetylation of histone. Acetyl groups is added to histone to make more gene being expressed and increase the possibility of the transcription of the DNA. And another example is the methylation of the cytosines or C base on the DNA. And this makes the DNA condense in a very tight structure, and so it prevents the transcription. So it simply deactivates the gene sequences. Detailed explanation of the methylation of cytosines. There is the loop of the nitrogen and carbon in the cytosine, and before it is methylated, on this carbon, it binds with a hydrogen and it forms a single covalent bond. And then when it is methylated, the hydrogen is replaced with a methyl group, or composed of the one carbon atom and 3 hydrogen atoms. So the functional group change. This results in the alteration in the bond angles of the DNA, and so it forms a condensed structure. In eukaryotes, mRNA is modified after the transcription process, and this modification is called post-transcriptional modification. And there is a reason for it. It is because of the compartmentalized structure of the eukaryotic cells. For example, there are nuclear membrane, and DNA is stored inside of the nucleus. And before the post-transcriptional modification, the mRNA is called pre-mRNA. And after this process, it is called mature mRNA. Here is a simplified diagram of the mRNA splicing, which is a type of the post-transcriptional modification. In the pre-mRNA here, there are both exons and introns. And in the splicing process, the splicing is done by spliceosomes. And in the final stage, there are only exons left in the mRNA, which this is called mature mRNA, and the introns is spliced out, and the spliceosomes simply break down. And there are actually other type of post-transcriptional modifications other than the mRNA splicing. And for example, this includes the adding a poly A tail, which is the section of nucleotides added to the end of the gene, and also the 5' cap.
So I'm talking about the translation. As we know, the gene sequence determines which kind of protein to be made by the cell. And after the mRNA transcribes the genetic information from the DNA and flow out of the nuclear pore into the cytosol, the information on the mRNA is going to be translated to synthesize specific type of amino acid. So in this topic, I'm going to cover the stuffs including the structure and function of the essential component, and the actual procedure of translation, and eventually the difference in the location of the ribosome.
First of all, I'm going to introduce the structure of the ribosome. As you can see, this whole ugly thing is a simplified diagram of ribosome. As an important organelle in the cell, its main function is to be a site for protein synthesis, which is to make a fantastic chain of polypeptide that could be hormones, receptor, transporter, or various type of enzymes. So the ribosome mainly got 2 subunits, as you can see, and you can just simply understand them as the large subunit and the small subunit. Also, it got protein and ribosomal RNA that do other complex jobs. What's most important about this is that it got 3 binding sets, and for TRA to bind, and they are the middle one is called the P site, is for the peptidosite, and the A site for the amino acid site, and E site, the left one, is called the exist site. Besides, there is also binding sets for the mRNA to bind. And later on in the video, you are gonna know what exactly they do during the translation process. tRNA, as the diagram shows, tRNA is a molecule that is composed of chain of nucleotide forming the loop structure, and it is what we call the arms. And as you can see on the diagram, the right arm of the tRNA is made of 7 nucleotides, as you can count on the diagram, and it is associated with the ribosome binding sets when it is conducting the translation. The left arm is what we call the D arm, which is made of 8 nucleotides, which is associated with tRNA activating enzyme before the translation occurs. The upper arm got 3 bases ACC codon for the attachments for the amino acid, and the lower arm, it got the triplets of bases is what we call the anticodons. And this one is what actually reads and translates the codons that are the mRNA to tell which kind of amino acid to be made in the translation. Since the different protein has its own designation for different purpose, the location of ribosome will be different. There are 2 locations of ribosome within the cell. One is the ribosome, those freely floats in the cytosol. As you can see, these are the free ribosomes. Another is bonded to the rough ER, and it is what we call the bound ribosome. Translation in the cell commonly happens in the free ribosome, and the protein synthesized from the free ribosome are mostly for the use in the cytosol, mitochondria, and the chloroplast. And the protein synthesized from the bound ribosome are mostly for the use of ER, the Golgi apparatus, and the lysosome, and outside of the cell. That's because that the rough ER can provide a vesicle that can transport the protein inside of it within the cell. Whether the ribosome is bonded to the rough ER or not is determined by the signal sequence on the polypeptide. As you can see on the diagram, the first parts of the polypeptide synthesis is known as the signal sequence, and it can be recognized by the sequence recognized protein. Once the signal sequence show up, the signal recognition protein will bind to it, and it will stop the translation until it reaches the receptor of the rough ER. As you can see, once the whole thing reaches the receptor on the rough ER, it will then start again the translation, and the translation will put the polypeptide synthesis into the rough ER. Therefore, after the synthesis is finished, after the translation is finished, the rough ER can give it the vesicle to transport it within the cell.
The translation is a series of events that happen in a certain order. The first step is called initiation. The small subunits of ribosome will first attach to the mRNA, and as the tRNA reaches the starting codon AUG, the large subunit will bind to the small subunit and fit the first tRNA into the peptidosite, the P site. Then the elongation will happen after the initiation. The direction of elongation is from the 5' to the 3' of the mRNA. Every new arriving tRNA will attach to the amino acid sites to read one of the codons on the mRNA to synthesize specific type of amino acid. There is maximum of 2 tRNA translating the mRNA at the same time. Meanwhile, the amino acid from the 2 tRNA will form a peptide bond with each other, and the dipeptide will keep attached on the tRNA in the A site. As the elongation continues, the previous tRNA will enter the exist site to detach from the ribosome, and the tRNA that was in the A site will move to the P site. At the same time, a new tRNA will again arrive and refill the A site to read one of the codons to synthesize a specific type of amino acid. This series of events will repeat as a cycle. Therefore, a chain of polypeptide will eventually be synthesized as more and more amino acids are synthesized and bonding with each other. The elongation will continue like this until the tRNA reaches the stop codon UAA. The stop codon will terminate the translation, following a breaking between the polypeptide synthesized and the tRNA. Besides, the ribosome, tRNA, and the mRNA will no longer attach to each other and then break down.